In this part of toxicity test, we're going to look at pharmacokinetics and metabolism. This episode toxicity test will be in two parts. In part 1 we will look at a paper Dr. Jill Langley wrote called Replacing Animals in Absorption, Distribution, Metabolism and Excretion Studies, which can be found on the AllTox website. Extracts from Dr. Jill Langley's paper will be intercut with clips from an interview she did with Peter Tatchell in 2007. And in part 2 we will hear from Dr. Ray Greek, Dr. Margaret Clockworthy and Dr. Katia Zayan. These next 4 slides are taken from the AllTox website. This first slide gives us a definition of pharmacokinetics. It reads pharmacokinetics stroke toxicokinetics. They usually say toxicokinetics when they're talking about chemicals and pharmacokinetics when they're talking about drugs. Anyway, it's defined as a study of the rates of absorption, distribution, metabolism and excretion, ADMER for short, of toxic substances or substances under toxicological study. Pharmacokinetic stroke toxicokinetic testing involves describing the bioavailability of a substance and its kinetic and metabolic fate within the body. Pharmacokinetics is also the term used to describe the assessment of ADMER in the context of drug preclinical testing. This second slide gives us a definition of metabolism. It reads, metabolism has been defined as all aspects of the fate of a substance in an organism. However, metabolism generally refers to the biotransformation of a substance via enzymatic or non-enzymatic process within the body to other molecular species, usually called metabolites. For ingested substances, metabolism primarily takes place in the liver, although many organs and tissues have metabolic capability. 2 types of enzymes are involved in metabolism. Phase 1, cytochrome P450 enzyme family, and Phase 2 enzymes. This slide describes the animal tests and the non-animal alternative methods. The animal tests reads, toxicokinetic studies are described by the Organization for Economic Cooperation and Development Test Guideline TG417. The route of administration, number of doses and animal species may vary with the purpose of the study. The substance and/or metabolites are determined in body fluids, tissues and/or excreta. Under non-animal alternative methods it reads, due to significant species differences in the types and activities of the enzymes involved in metabolism, this is one area where human-based models are critical for protecting human health. Many types of in vitro methods and test systems have been developed to assess metabolism. The advantage is that models using human cells provide species-specific results. A major disadvantage has been the rapid loss of metabolic enzyme activity by cultured cells and tissues. Specific assays and approaches for the non-animal assessment of metabolism have not been validated, but they are widely used by industry given the need for human data. In this 4th slide it talks about in vivo human testing, and it's specifically talking about microdosing, which has been used to evaluate drug ADMER. However, it says this approach has not been widely accepted for testing chemicals. The title of the second paragraph is Validation Acceptance of Non-Animal Alternative Methods, and it reads, the Interagency Coordinating Committee on the Validation of Alternative Methods, which is the USA Centre for the Validation of Alternative Methods, ECVAM, which is the European Centre for the Validation of Alternative Methods, and the OECD have not yet reviewed or endorsed as scientifically valid any non-animal methods for evaluating metabolism and pharmacokinetics stroke toxicokinetics.
But the problem scientifically with using other animals is that while clearly there are similarities between humans and other animals, there are also many differences. And these can be at the level of the organs in the body, which may have different structures. They can be at the level of the tissues or the cells. There can be molecular differences between the species. There can be differences in the way the circulation works or the nervous system. And so when you do an experiment on an animal, you can never know for sure whether that will predict what's going to happen in people. And oftentimes it doesn't. And that is a really serious problem.
So here we are looking at Jill Langley's article called Replacing Animals in Absorption, Distribution, Metabolism and Excretion Studies. The first paragraph reads, "The absorption, distribution, metabolism and excretion (ADMER) characteristics of chemicals are central to interpreting hazard data and making risk assessments. And in pharmaceuticals, the ADMER properties of medicines are critical to their success or failure. Improving human welfare and gaining economic advantage are powerful drivers for accomplishing the best possible ADMER studies with drugs and chemicals. And of course there's a third equally important motivation, preventing animal suffering." It goes on, "Until tests for ADMER can be achieved without using animals," that is debatable whether they can be achieved using animals now of course, "it will be very difficult to replace a range of systemic toxicity tests on animals. Progress will also be delayed in advancing in vitro and in silico methods from the status of screening tests to full replacements. Therefore, predicting human ADMER more effectively and without using laboratory animals is a pivotal goal."
And in fact, the world's largest drug regulatory agency, the American Food and Drug Administration, has concluded that 92% of all the drugs that pass animal tests are either ineffective or toxic in humans, which means that the drug industry is working with the animal experiment system has only an 8% overall success rate, which is just catastrophic. It's catastrophic for animals' lives and it's catastrophic for people too.
In this second slide, the second paragraph reads, "In human health studies, including assessing the ADMER characteristics of xenobiotics, animal testing has been placed at centre stage since the 1960s. Early on, it became clear that animal-based toxicology had severe limitations. In 1980, for example, Professor George Teeley Smith summarised the situation for pharmaceuticals. Professor George Teeley Smith said, and remember this was in 1980, "There is at present no hard evidence to show the value of more extensive and more prolonged laboratory testing as a method of reducing eventual risk in human patients. In other words, the predictive value of studies carried out in animals is uncertain. The statutory bodies, such as the Committee on Safety of Medicines, which require these tests, do so largely as an act of faith rather than on hard scientific grounds." And this new slide just carries on from the last slide and reads, "And for chemicals, the British Royal Society has reached a similar conclusion at the same time. Unfortunately, this use of animal models for predicting risks for man is beset with difficulties. It is rarely possible to be sure that the animal model properly represents the relationship in man. Even if it were possible to improve the accuracy of the present-day test procedures so that the risks to the test animal were known with greater precision, this would not necessarily bring about a corresponding improvement in the assessment of the potential risk to man because of the uncertainty regarding the relevance of the animal data for man." Dr. Jill Langley's article then goes on to suggest that it's time for change. The second paragraph reads, "Everyone knowledgeable in this field is aware of the deficiencies of animal testing, but a familiar system is more comfortable than change. Traditional toxicologists and regulators have ploughed on, arguing that the animal-centred approach is the best for the time being, even in the absence of systematic data on the validity of animal tests. They have tried to adapt the tottering edifice of regulatory toxicology to each new challenge, and in recent years there have been plenty. In chemicals regulation, the challenges include endocrine disruptors and nanomaterials. In medicine, an example is the safety testing of therapeutic proteins, brought starkly to light by the British disaster with the humanised monoclonal antibody TGN-1412, which nearly killed several volunteers in clinical trials conducted in March 2006. Dr. Jill Langley's essay then goes on to say how things are changing. This slide is titled "Replacement Goes Mainstream." It reads, "It is only in the last few years that non-animal toxicology has made a genuine breakthrough into mainstream science. Increased support at European Union level, together with legislative initiatives and visionary reports from highly influential agencies, have combined to take the approach to a new level. The work of the European Centre for the Validation of Alternative Methods has consolidated the strategy and produced a number of validated non-animal assays. In 2002, the European Union's 6th Framework Programme on Research and Development offered unprecedented opportunities for funding to replace animal-based toxicology. More than 20 research projects, some of them tackling the ADMER properties of xenobiotics, were funded to a total level of about €92 million. If you read through, you can see acute tox, which was mentioned in an earlier episode of toxicity tests. The second paragraph talks about changes in legislation. It reads, "Legislative initiatives have also been highly significant in sharpening the focus on non-animal methods. The 7th Amendment to the EU Cosmetics Directive, which introduces bans on the animal testing of cosmetics ingredients, has stimulated new research efforts." There's a spelling mistake there. The REACH legislation included intelligent testing strategies, incorporating non-animal methods which have minimised the number of animals to be used in the REACH testing programme.
Why don't you think the government is giving a lead?
I think the government has completely lost its way on the whole issue of animal experiments. There's a policy vacuum at the centre of their engagement with the issue, and there's no leadership at all. The funding that this government puts towards developing non-animal methods is embarrassing in international terms, and some other countries like Germany and Switzerland are way ahead of us on this.
What about the European Union?
The European Union is coming round to the idea, and I think at the level of Europe we might actually be on a bit of a cusp scientifically. The Commission of the European Union has funded a number of international research projects, getting scientists together from different countries with different expertises to try and find ways to develop new tests for drugs and for chemicals that don't use animals. So I'm quite encouraged that the European Union is starting not only to speak the right language and to acknowledge that non-animal testing is advanced testing that's more cost effective and reliable, but they're actually putting their money more where their mouths are.
Jill's article goes on, "A major step was achieved with the publication of the US Food and Drug Administration's 2004 Critical Path Report on medicines development. This highlighted how the current drug development process is increasingly difficult, costly, and inefficient. The report identified as a major problem the failure to create and use novel scientific tools to deliver fundamentally better answers about how the safety and effectiveness of new products can be demonstrated in faster time frames with more certainty and at lower costs." The second paragraph reads, "In 2007, another seminal report emerged, this time commissioned by the US Environmental Protection Agency. The EPA recognized the need for a full review of toxicity testing frameworks for drugs, food additives, pesticides, and industrial and other chemicals, and asked the National Research Council to conduct this review and propose a new vision and strategy for toxicology. A key feature of the proposed vision is to move towards human-relevant in vitro and computational techniques that offer faster, more efficient, less expensive, mechanistically based and more comprehensive assays. As the NRC report said, toxicity testing is approaching a scientific pivot point. It is poised to take advantage of the revolutions in biology and biotechnology. Advances in toxicogenomics, bioinformatics, systems biology, epigenetics, and computational toxicology could transform toxicity testing from a system based on whole animal testing to one founded primarily on in vitro methods that evaluate changes in biologic processes using cells, cell lines, or cellular components, preferably of human origin.
I've heard myself, scientific advisors for pharmaceutical companies saying, well,
yes,
we would love to move to non-animal tests
yes,
because they're more efficient, more cost effective,
yes,
but we've got this barrier that says these drugs have to be tested on animals.
Yes, although if you listen to the drug companies and you listen to the regulatory agencies, they usually blame each other. So the regulatory agencies say, oh, well, we would be very interested to look at data from non-animal tests, but the drug companies don't provide enough of it. And then the drug companies say, but the regulatory agencies won't accept our non-animal data. So there's a bit of circularity there and clearly some inertia that needs to be overcome. And if only governments would give a lead, then I think that circle, that little vicious circle of blaming each other could be broken out of.
This slide just talks about how much data already exists in the world on drugs and chemicals. It reads, the pharmaceutical sector has much more experience of in silico and in vitro strategies than does the chemicals industry. It also has the advantage that during drug development, molecules are assessed in non-animal, animal and human trials, providing a huge potential data resource which should play an essential role in validating novel approaches. Drug regulatory agencies such as the US Food and Drug Authority and the European Medicines Agency hold parallel test data for hundreds of novel drugs. Data mining will inevitably reveal information of direct importance to the development and validation of non-animal techniques to assess xenobiotic ADMER, as acknowledged by Sir Michael Rawlins, Chair of the British National Institute of Health and Clinical Excellence. He says, clarity can be provided by analysing the hundreds and hundreds of studies locked in the files of the regulatory authorities in both the US and the EU. In particular, it is worthwhile looking at drugs that failed or have been withdrawn. This slide again suggests that hopefully things might be changing at the regulatory agencies. It reads, in the pharmaceutical arena, the US FDA acknowledges the substantial limitations of animal testing. Its Deputy Commissioner for Operations, Janet Woodcock, has said, we believe the applied sciences have not kept up with the basic science in drug development. Therefore, when discoveries made using advanced sciences are brought up to the preclinical stage, that's the animal testing stage, we start applying technologies that have been used for 50 or 60 years, such as animal tox, which is completely empirical. We just dose the animals and cut them up and see what happens.
There you're referring to the regulatory agencies, both in this country, in Europe and around the world, who insist that some animal testing is done. So there has been considerable inertia in those regulatory agencies who seem to be happy to plough on with the same animal tests that were being conducted 60, 80 and in some cases nearly 100 years ago. So some of these animal tests are incredibly outdated. And I even read about a very high up person in the American Food and Drug Administration who said, basically, we're doing the same drug testing that we did in animals 80 years ago, put the drug in them, kill them and cut them up and see what happens. That's not 21st century science. So there has to be a shift to the more advanced non-animal methods.
This slide just says how the FDA is also recognising computational modelling.
There's also some wonderful new developments in terms of computer simulation, where you can do virtual experiments on a computer that don't require you to have any of a drug. You don't have to have synthesized your new drug even. You just can have a look at the molecule as an image on the screen and see how it might react in different parts of the body. So there's lots of exciting new developments there as well.
So you're also alluding to the new generation of virtual reality supercomputers, which can actually model
Yes.
whole organs, even whole parts of the body.
Yes, that's an up-and-coming area, and the supercomputers are absolutely crucial to that sort of work. And there's also some exciting developments in cell culture, because cells have usually been grown in very simple test tube circumstances where they don't really resemble the way they grow in the body, but increasingly scientists are finding ways to almost like make mini cellular organs in the test tube that much more resemble what happens in the human body. So they're combining different cells together and recreating structures that you would find in the human body, and then being able to do experiments on those in the test tube.
Continuing the theme of recognising alternatives to animal-based methods, this slide says, one way to obtain gold standard human ADMER data is by means of microdosing studies in human volunteers. Increasingly utilised by the pharmaceutical industry and backed by recent guidelines from both the US FDA and European Medicines Agency, microdosing involves giving a sub-pharmacologic and sub-toxic doses of a test compound to volunteers. New techniques of analysis that are extraordinarily sensitive, such as accelerator mass spectrometry, allow the measurement of ultra-low doses of new drugs and their known as well as unknown metabolites. PET scans can be used to ensure a drug microdose has penetrated to the target organ, e.g. the brain. Microdosing can preempt some animal tests and reduce the cost and timescales of drug development through predicting early in the development process which drugs will ultimately fail in humans. If it fulfills its early promise, human microdosing will also provide data in the species of ultimate interest to assist in the validation of new in vitro and in silico ADMER techniques. Dr. Jill Langley finishes her article on ADMER with this paragraph. It's titled "A Pivotal Moment." We are witnessing an exciting and challenging paradigm shift in toxicology, a pivotal moment when non-animal techniques are going mainstream. There is a confident expectation that this will benefit chemical hazard and risk assessment as well as drug development. Novel tools applied intelligently in a 21st century toxicology framework will help save time, resources, and human and animal lives. Such a win-win solution should be pursued with the maximum enthusiasm, support, and leadership from the scientific, regulatory, political, and animal protection communities.